Okay, well, we'll get started. Sorry, I'm such a creature of habit. I just went straight to Beckman and nobody was there. It was thank goodness Albert sent me a text. So I guess I would have figured it out eventually, but thank you to the whole VP team for organizing all of this. This is really fantastic. It's exciting for us to see this workshop. We've been talking about doing this kind of thing for quite a long time, and there's so many outstanding workshops out there. So it's tough to find an area that's unique and I think valuable to students. So we're really interested in your feedback on that. I know everybody does want to have bicycles and surfboards for the future, for the next time, so that would help it a lot. And thank you all for coming. So and being the kind of guinea pigs in some sense for the very first one. We're excited to have you here, and this is the so tomorrow is. What is the topic tomorrow?
Wide field imaging.
Wide field imaging, okay. Yeah, and so Sylvan will talk about that? Yeah. Okay, excellent.
And Carol will talk about inverse problems.
Okay, fantastic.
Both spectral as well as...
So these 2 days will deal mostly with multiply scattered photons, deeper penetration, and we'll try to, today, give you a good foundation in some of the conceptual basis for those methods. And tomorrow you'll have even more expansion on that. And you'll be able to contrast between techniques that use probes that are in contact with tissues versus imaging cameras and how to get quantitative information. So this is the motivation talk, the one to hopefully get you excited about what you can do with diffuse optics. And what I'll talk about really is medical imaging. I don't think I have any small animal preclinical model imaging in this talk. So this is all about people and what we can do with these technologies. So how many of you actually think medical imaging with diffuse optics is like a big thing? It's happening all over the place. Okay, good. It's it's not one of the mainstream medical imaging modalities. It is in some sense ubiquitous. And we'll talk about that. But it's not considered to be mainstream. Who controls the mainstream medical imaging technologies? I'm thinking of one particular community. And I'm not talking about scientists. I guess that ultimately they control everything. But radiologists are essentially in control of medical imaging. And kind of the basic technologies that we have in radiology will be MRI, X-ray, ultrasound. Will this actually work?
Advanced slide.
Yeah? Oh, yeah. So it's got a USB there. Okay. So X-ray techniques, MRI, ultrasound, nuclear imaging methods. So optics is kind of set off to the side. And if you think about optics, and I thought I had maybe, I guess I took this slide out, but what's supposed to go there are the mainstream modalities, the X-ray, the MRI. So optics is kind of set off to the side. It's a developing technology in a lot of ways. It's not in that same area with those mainstream methods, but it's also everywhere. It's used as a monitoring device. It's used as an anatomic imaging device. So here's some examples of standard of care with optics. In the eye, I think everybody's familiar with OCT, optical coherence tomography. It's become the standard technique. It's the fastest growing technology in ophthalmology. It's probably the most transformative one to be introduced over the last 20 years. And currently, it's able to provide really beautiful images of every retinal nerve fiber layer. So this is a cross-sectional view from a commercial instrument of the retina, and you can see very nice contrast with nerve fiber layers. It's standard of care. There are, 7, 8, 9, 10 companies that are developing OCT technologies. It's really an application in the eye that's really very well matched with the technology. And emerging aspects of OCT that are still more or less anatomic are looking at Doppler shifted signals or speckle-based imaging in OCT, where essentially you take the scattered light and you form images from moving structures. So moving structures in tissue will be erythrocytes. And you can form images of blood vessels. This is mouse cortex. This is done by Zhongping Chen here. So this is a type of Doppler OCT. It looks at the Doppler shifted scattered signal. And from mathematical reconstruction techniques, you can visualize vasculature. And depending upon how quantitative you want to be, you can determine whether it's going in or out of the plane of the field of view. You can even measure the actual velocity. That's tougher, much tougher to do. So right now it's being used more or less as an anatomic contrast imaging approach, looking at the basic structure of blood vessels, the tortuosity of blood vessels, trying to understand the vascular density in different tissues. And in particular, there's a lot of interest in doing this in the eye. So many diseases in the eye. Can anybody name some diseases in the eye, pathologies in the eye, where it would be beneficial to have a combination of retinal nerve fiber imaging as well as vascular imaging? What's the most common cause of blindness in people over the age of about 50? Well, I think all those are pretty big things. So those are good. So there's a lot of interest in macular degeneration. And certainly diabetic retinopathies are really crucial issues. There are alterations in the vasculature, and they affect people of all ages. Glaucoma is not so much a disease of overvascularization, and some aspects of glaucoma are still mechanisms or physiologic mechanisms are still unknown. But what accompanies it, of course, is intraocular pressure. That can cause compression in the nerve fiber layer and diminished perfusion in the choroid and in the retinal vessels, for sure. But macular degeneration is a really big problem, particularly in the aging population. There's a lot of interest in applying OCT and related technologies to detecting it earlier and looking at the response to therapies. And endoscopic techniques, everybody's familiar with these. So they're more or less standard of care everywhere. Surgery is less and less invasive and more minimally invasive with optical guidance. And there is a lot of activity to try to introduce more sophisticated tools to give information, feedback to surgeons on margins, on the efficacy of surgery, where they should cut, how much they should cut, and so forth. So these are all standard anatomic imaging techniques. They provide high contrast information. So when you see an image, you kind of recognize the anatomy. These methods, functional methods, typically if they do form images, they're not high-resolution anatomic images. They're all based on measuring some aspect of tissue function, and typically it's some relationship between cells and vasculature. So cells need glucose, they need oxygen, vasculature provides it. As cellular metabolic demand increases, there is an accompanying vascular change. You may have a tissue like adipose tissue that's not particularly demanding, and there won't be lots of vascular changes. And so that's may not be as interesting to study in a dynamic setting. And then you may have tissue like brain that's obviously quite demanding, and alterations in metabolism, cellular metabolism, will immediately impact the vascular response in the brain. So here are two examples of standard of care technologies that are functional. One is pulse and hemoglobin oximetry. This is a company, Massimo, that's actually here in Irvine. They have the largest market share in pulse oximeters. They've also developed a device, and it's actually this device, the Pronto, that measures hemoglobin in the blood non-invasively. So it's a spectroscopic measurement that uses the same principles of pulse oximetry, but with many, many wavelengths to try to deduce the concentration of hemoglobin using a totally non-invasive sensor. So that's done now. The clinical standard of care is pretty simple. You take the blood out and you analyze the hemoglobin content in a laboratory. This is a device made by a company called Somanetics, which was then purchased by a company called Covidian. Have any of you heard of Covidian? Is everybody a biomedical engineering? So Covidien could be one of the largest biomedical engineering device companies, biomedical device companies in the world. So you should hear of Covidien. You may get a job with Covidien, or you may start a company that rivals Covidien. So it's important to know what they do and the whole range of technologies that they develop. This one is not something that Covidien actually developed. They purchased it from a company called Somanetics, or they bought the company, Somanetics, which pioneered the development of this INVOS oximeter, which is simply a CW multi-wavelength oximeter that's primarily used for brain monitoring. Does anybody know where the biggest applications of that are? What part of the hospital could you walk into and see one of those devices? In almost any hospital in the US. I don't know about outside the US. They have better technology outside the US. Any guesses? Where?
Wherever babies are.
Wherever babies are, exactly. So neonatal intensive care units, pediatric intensive care units, they call those NICUs and PICUs. So almost every baby is hooked up to one of these. How come? Let's say it's neonatal intensive care. We actually kind of touched on this yesterday when I was talking about the oxygen analyzer. You didn't know you'd have to work so hard this morning. So why would you want to put an oximetry probe on a baby's brain, especially premature? But why would it not be functioning?
Well, I mean, since they're premature babies, they're maybe underdeveloped in some ways.
Lungs.
Yeah.
So that's a key issue. Lungs are among the last organs to develop. And so when they're premature, the lungs are not well developed. That's why they go in incubators. And that's why you put them, so you can control the oxygen tension very carefully. And you want to make sure that they're fully oxygenating their brains, because if you don't have adequate brain oxygenation, then there's no brain development. And if you don't have brain development at those early stages in life, then you end up with major problems. So this is a key issue. And how could that potentially impact clinical decision making? Why do clinicians like that? So you've got a probe on a baby's brain who's got immature lungs, and then what could you do? You are now the neonatal intensivist.
Measure oxygen content in the blood and brain.
I guess you'd have to have this one for the blood and this one for the brain. And so you can follow that over time, and you can see what types of fluctuations there are. What if the baby stops breathing? Well, you'll have other sensors that tell you it stopped breathing, but you could see what the impact of that is on oxygenated hemoglobin delivery to the brain. And you could intervene if you see that the saturation levels are too low. What if there's an injury during birth, or there's congenital deformation in one of the hemispheres of the brain? What if there's a blood clot? Something happened? What could you tell with this device? Well, I'll answer that one. So you could tell if the tissue is injured, if it's metabolically active, if it's repairing. So this can give you lots of dynamic feedback, and it's a trend monitor. So it's just put on the infant, and you have it as part of the monitoring station, and the nurses can monitor it. You can monitor it on your cell phone. You can get information that's transmitted to you. So it's a very powerful monitoring tool. So we'll talk about how good these things are, but also where their weaknesses are and how to make these better. And so Albert and I are going to kind of team up on that and give you a sense of what you can do with these technologies and what is the theory that you need, the conceptual framework that you need to make these things work even better. So the next generation technologies, those are the ones I just talked about, are pretty good. In functional imaging, we have a tough problem in some sense, like from looking at things, because the anatomic imaging technologies are so beautiful, but they're high resolution, relatively small field of view, and specialized to that particular anatomy that you're looking at. Whereas the functional technologies, we're trying to see as deep as possible beneath the surface and integrate information up over a large volume. We're, from a research point of view, very active in trying to understand what the biologic origins of contrast are. So when you see the signal go up or go down, you want to know what is the driving biology behind that. And that's one of the unique features of functional optical imaging, in that what we measure with optics, we can relate across spatial scales to the other technologies that you've heard about and that you know about. So those can provide us even better, more powerful insight to the underlying physiologic and metabolic changes that are ongoing in disease. And ultimately, what we all like to do, and something, for those of you who are biomedical engineers in particular, you want to always be thinking about how to link an endpoint, some measurement that we make, to a clinical outcome. So if you are involved in clinical and translational research, it's great to explore stuff, and we want to do that as much as possible. But we also want to have at least a few things where we're really pushing the envelope, we're testing ourselves. It's our kind of like our stretch goal. And that's to, if we see something go up or go down, quantify it, and then say, here is a predictor of how the patient will do. If we can measure this, we can intervene, and the patient will turn out better. So that's a really, really important goal that we shouldn't lose sight of as we're developing all of these technologies. So tissue optical imaging, here's the big picture, and you're getting the big picture this week. They're just a couple of, it's a simplified big picture, and there are just a couple of features I want to point out in this roadmap to help orient you as to where we are. On the y-axis, we have resolution, and the x-axis, we have depth. This is not a quantitative picture, it's a cartoon, it's a schematic. But broadly speaking, these blue technologies, nanoscopic or subwavelength imaging, microscopic techniques, coherence tomography, these all treat light as a wave, and they require the rejection of multiply scattered photons. So you need to have coherent information content in order to form images. And there are various strategies that each of these use in order to form coherent images. So coherence tomographies typically use interferometric gating in order to suppress multiply scattered light, light that does not have relevant information. Microscopy techniques use a variety of gating strategies. They can be a simple pinhole, like in confocal microscopy. They can be electronic selection, electronic gating, such as in nonlinear microscopies. Everything in these methods is designed to take advantage of optical phase relationships. And they're high-resolution techniques. If you bring them into tissues, they're anatomic methods. Of course, you can combine them with exogenous probes, and they can be more functional. But in general, these are anatomic or structural techniques. And once light goes about 1 millimeter in tissue, which is what we call the transport scattering length, then all those optical phase relationships become stochastic with respect to one another. They're hard to tease out. They're varying so much in space and time that it's very difficult to pull out coherent information content. Now, there is a lot of work to try to localize light at depths of about 1 millimeter and do techniques such as time reversal techniques. And so there are many state-of-the-art methods that are under development at this interface in sort of the millimeter dimension to try to get coherent information content. But the bottom line, as Albert will show you, is there are very, very few photons that are available to form images. So the overwhelming number of photons are what we call multiply scattered. And, why fight it? There are a lot of them available, and there actually is great information content in them. And these are these diffuse techniques. And if we're keeping track of light, then we need to do bookkeeping in the form of a transport equation or a diffusion approximation to a transport equation, and think of light as a particle rather than as a wave. So this is kind of a nice representation. You have optical methods that are based on a conceptual framework of light as a wave, and we have techniques that penetrate more deeply into tissue, where optical phase relationships are stochastic. You can't really squeeze coherent information out. So we need a conceptual framework that treats light as particles, and that will be the transport equation, and you'll hear lots more about that. And you already have heard a lot about that. And these methods are macroscopic imaging techniques, photoacoustic tomographies, and diffuse tomographies. One of the important things about this is that even if I'm using a diffuse tomography and I'm looking at fluctuations in some light field that's interrogating several centimeters in tissue, I'm looking at scattering. So where does scattering come from? Did you guys have that lecture? Where does it come from? Like from a very physicsy point of view. Excellent. Index mismatches. And so what's causing the index mismatch? No, from a biology point of view. Like a top one, one of our favorites is mitochondria. It's about a micron. It's high index, double bilayer, super scattering. What's another big scatterer in tissue? Collagen, extracellular matrix proteins. They assemble, collagen fibers assemble on a spatial scale that's about the wavelength of light. Super strong scatterers. What's another extracellular matrix protein? Kind of typically goes along with collagen. Collagen gives, some rigidity to the tissue, some stiffness. What's one that imposes some elasticity? Elastin. Yeah. So those are pretty big structures. They scatter light very intensively, and our scattering signal is revealing information about those cells and extracellular matrix structures. So those diffuse techniques are related to coherence methods. The information content that's embedded in them is related. And of course, one of the big challenges of our field is to devise studies that allow us to acquire information across spatial scales. And just, I'll plug some work that we've been doing with Vossen and Carroll. I think a good example of that is a recent publication that we had in Biophysical Journal looking at single cell metabolism in skin, keratinocytes, which we looked at using non-invasive multiphoton microscopy to look at intrinsic NADH signals. And then we were able to model where the light went and where the signal came from using these techniques that you guys are learning about that take advantage of multiple light scattering and structural properties. And we combine that also with a spatial frequency domain imaging technique that Sylvain will talk about to look at oxygen consumption and metabolism. We put all that together, mixed it up, and we were able to determine the rate of oxygen consumption per cell per second in the skin by keratinocytes. So there are challenges out there like that. If you're interested in those types of things, how you fuse these technologies across spatial scales together to get really fundamental information that could potentially be useful clinically, take a look at that paper. And there's not enough. It's really hard to do. But when you start to get involved in it, I think it can be very satisfying. And there are a lot of opportunities there to kind of fuse that information across spatial scales. So today we'll just talk about this spatial scale, diffuse tomography, and what kind of information you can get from it. But I'll always be referring back to the microscopic scale and inferring from our signals what's going on with cellular metabolism. Because ultimately, it's cellular metabolism that's driving the vascular changes that we're interested in measuring. Okay, so this is very real world. It's not so abstract. This is bringing you back to reality of what is multiple light scattering. So Albert took this picture of me. It's a Canon Rebel camera, and the only modification that we made to the camera was that the IR blocking filter was removed. So even the Bayer filters are on this camera. And I'm being illuminated from within by 850 nanometer near-infrared LEDs. So there's no outside light source. Everything is dark. The only light that's on are these LEDs, and they're stuck in a little device with a battery, so it's totally handheld. And you can see there's a lot of light. So this really illustrates, first of all, it's blurry. So you have no idea. We put this little barrier up over here because the light leaks out of your mouth. And you could see that this is where the source is. But when I block it, if I didn't tell you the source was here, you'd have no idea where the source is. So this illustrates the point of randomization, multiple light scattering. You lose track of where the light started in the first place. It also illustrates a really nice feature. What's the good news about this picture compared to other medical imaging technologies? Have any of you worked with MRI? How many of you think MRI is the most impressive technology ever? I think it's pretty impressive. But what is the weakness of MRI? Actually, there are a lot of weaknesses to MRI. What about like from a hardware point of view, an actual measurement point of view? Forget the fact that it's $1 million per Tesla.
Super huge, not portable.
Yeah, those are easy targets. What's a dirty secret of MRI, like that no one ever talks about? Signal to noise. You're practically in the noise floor. You're always battling signal to noise. And how many of you guys consider yourselves to be measurement people, instrumentation people? I hope some of you will raise your hands on modeling, because Boston would like to see that. And modeling and instrumentation go together. But if you're building instruments, your first job is to get the noise floor down as low as possible. And you know that GE and Philips and everybody else, and Siemens, they've worked really hard on their systems to get the noise floor down. Nevertheless, they're working in the noise floor on those techniques quite often. Where is the noise floor with this method? I don't know. This is pretty good. There are a lot of photons. There are tons of photons. So that's the good news. So that's one check, good news. Bad news is we have no idea where they came from unless I told you. So you're going to have to solve some crazy inverse problem unless you have prior knowledge, like I told you that this is where the light came from. Second is that, I think that that's probably bad enough. You have no idea where the light came from. So there's a lot of light, but you have no idea exactly where it is going or its direction. There's randomization and so forth. So that's the big challenge of our field, is to try to reconstruct where the light has been and then extract information content. And it's very seductive. Like you look here and you say, I would like to say that this is telling me that I have a sinus infection right there. So it's incredibly appealing. But we don't really know for sure unless we know that the light that's emerging from this part of my face actually really and truly did interrogate that sinus. So that's one of the big challenges of our field. And we call those inverse problems. And there are different levels of inverse problems. I guess we're not really going to talk so much about... Carol, will you talk about inverse tomographic imaging problems? Okay, so you'll have to come back for that course. So the first level of inverse problems really is not necessarily to figure out exactly where the light has been in space, but to figure out the relative amount of time that it's interacted with absorbers and scatterers. So if we can figure that out, we can determine the distribution of absorption and scattering in tissue, and then we can quantify the composition of the tissue. So that's doing non-invasive biochemical analysis. And if we can do that, there's a big payoff. So the payoff is in the spectroscopy. And the spectroscopy means the wavelength-dependent aspects of light propagation in tissue, or interaction between light of different wavelengths and the tissue itself, so that we can get information out. So just as a kind of a summary of what that information is, these are the main absorbing features in tissue from 400 nanometers out to 1,000. And these are their structures. melanin, which doesn't have any particular electronic structure. It just falls off with wavelength, both eumelanin and pheomelanin. It's a very, very strong absorber and strong scatterer. And we have deoxyhemoglobin and oxyhemoglobin. They have characteristic peaks in the visible and in the near infrared. And then there's the wavelength dependence of scattering. And so the key thing to point out here, why do we have multiple light scattering? It's because in the bluer wavelengths, bluish-green area, absorption and scattering are about equally likely to occur. And then as we move further out into the near infrared, absorption really takes a dive. So between 600 and 700 nanometers, we see that almost 2 orders of magnitude loss in extinction coefficient for hemoglobin. So that's a significant reduction in absorption. That's kind of the first part. The second part is that the dominant scattering particles in the blue are very small molecules. So this is Rayleigh scattering. And the wavelength dependence is 1 over lambda to the 4. And then as we move further out into the near infrared, into the red and near infrared, the main scattering particles are actually larger. They're on the order of the dimension of the optical wavelength. So those are those refractive index discontinuities, but they're actually more or less well-defined structures, like mitochondria, like collagen. They're about a micron. And so now the wavelength dependence of scattering flattens, and it's 1 over lambda. So you can just see that scattering is much more likely to occur than absorption in tissues. And this is the origin of multiple light scattering. And so in this window where there is lots of scattering and not as much absorption, we have really nice spectral features. So if we can do the first inverse problem, solve the first problem, which is figure out the relative amounts of time that light has spent interacting with a scatterer versus an absorber, then we can separate scattering from absorption and determine the concentrations of deoxyhemoglobin, which has a really beautiful spectral feature here and a very strong absorption below 800 nanometers. Oxyhemoglobin, which has an isospestic point, a point of equivalent molecular extinction at about 800 nanometers. And lipid, which has a nice peak at around 920, and a little shoulder that actually tells us something about the saturation of the fatty acid. And water. So water has very nice absorption starting at around 960 up to about 980 or so. These lipid and water peaks are harmonics of infrared vibrations. So they're overtones. They're not the same as these electronic absorption states. So these are movement, these are due to the movement, these features are due to the movement of outer shell electrons from highest occupied molecular orbitals to lowest unoccupied orbitals. So they're single electronic motion. These are vibrational features or a consequence of vibrational features. So what is the first kind of principal difference that you might expect in information content between these guys and the electronic features? That's a tough question. In general, can I count on the position of all of these features? Which ones are going to be more reliable when I make measurements? The spectral shape and location. The ones in the near the ones further out above 900, or the ones that are probing electronic states? How many say the ones probing vibrational states? How many say the ones probing electronic states? Exactly. Because these are overtones of fundamental vibrational states. They're recombinations of bending and stretching modes. So the molecules, or the moieties within the molecule that are contributing to those states, can be dramatically influenced by association with other molecules. So you get anisotropic vibrations and anisotropic recombinations. So those features can change in shape and in position. And that's potential useful information content for us. I won't talk too much about that, but one of my former grad students, Sophie Chung, who's at Penn, did her entire thesis on looking at this water state and the factors that contributed to its movement, its broadening of the band and changing the position of the band. And that's kind of actually a really famous problem that people have looked at over many, many years. What happens when water associates with macromolecules? What type of water is that? Bound water. It's bound water. So water that's bound to proteins and other macromolecules actually behaves a little bit differently spectroscopically than water that is mostly unassociated. And you can imagine that could be potentially a diagnostic or an indicator of certain pathologic states of tissue. So it's a very important thing to look at, and MRI uses this to look at diffusion of water, so water penetration through barriers and association with other structures. So it's a very practical way to use spectroscopy in tissues. So our key challenge then, we want to be able to quantify these molecules in tissue. If we're going to do that, we need to measure the path length to measure their concentration. Because you can see with this multiple scattering picture of me that obviously the light has quite a long path length. We don't know what that path length is. That's one of the big challenges. So this is just your familiar cuvette view of that same picture of me. If I were totally transparent, then this is what the light would look like as it came through me. So think of the human as a cuvette. And if you're transparent, then you just use Beer's law. You know what the path length of the cuvette is. That's a centimeter in a standard cuvette. And then you can calculate concentration by knowing the extinction coefficient, and you measure the absorbance. If I have a real tissue, I have no idea what the path length is. The light's scattered multiply. It could travel really very, very far, tens of centimeters. And there is both absorption and scattering loss if I have a detector in any location. Let's say I have one on the other side. So I can't measure, I don't know what the path length is, so I can't, even if I measure an absorbance, or what I think is absorbance, or transmittance, I can't relate that to scattering unless I know what the actual path length is. Now, I would say that most people ignore path length, and they make those measurements anyway. But I want to really try to get these things embedded in your heads in some sense. I'm not saying everybody has to measure path length, but you should be aware of it, and maybe figure out clever ways to overcome it as you make these measurements, or just go ahead and measure it directly. There are a lot of techniques available. So what is the deal? Why should we measure it? From a physics point of view, the big advantage is we'll be able to separate absorption from scattering. We can determine B, so we'll have a transmittance or an absorbance measurement. We'll know our path length. If we know our extinction coefficients of those molecules that we're interested in, we can determine concentration. Then we can potentially localize information in 3D. We need to solve some inverse problems, but that is the next step. The first step is to measure the path length. So then potentially we can do tomography. From a physiology point of view, what's the big advantage? Well, if we can determine the concentration of all those molecules I just talked about, then we can measure perfusion and metabolism at depth, oxy and deoxyhemoglobin, and we can do that in different regions of the tissue where normally you wouldn't have access, or you'd have to stick a needle in to get access to it. And we can determine the concentration of other absorbers and fluorophores, so like I mentioned, water, lipid. I can put in exogenous dyes or particles. Are any of you guys working with exogenous fluorescent dyes? Really? Sivan? Yeah? So those are good. I mean, we're trying to develop instrumentation that's ready. It's a little frustrating to see everybody working on dyes and particles, and really nothing is clinically approved. It's going to take a very long time for that type of stuff to happen. But there are some that are clinically approved. So grandfathered in are indocyanine green, methylene blue. So those are used widely. They're used in medicine every day. Fluorescine. So you can determine, you can be more quantitative even with those ones that exist if we can do path length, because we can correct fluorescence for tissue optical property distortions. So remember, tissue optics, both contains information about intrinsic physiology, but it also distorts information content. So as an example of that, let's say we're trying to quantify fluorescence in tissue, and I have some intrinsic optical properties, some absorption spectrum and some scattering spectrum. And let's say this is the spectrum of my pure fluorophore. Once I put it in tissue and I measure it, after the fluorescent light has interacted with all of these scatterers and absorbers, it's like having a filter. So it will selectively attenuate in regions where there are intrinsic properties. And I may get some spectrum that looks like that. So if I know what my intrinsic properties are, then I can literally correct that spectrum. And so many people are now recognizing this and trying to do it. And in the process, what you get from that correction, not only can you quantify the fluorophore and the absorbers and the scatterers, you can also deduce where the fluorophore is located. So these are rapidly moving and emerging types of approaches. And actually, all the computational methods that you guys are learning put you in a position where you can do exactly that. So path length, everything is about path length. And so I want to now go through a little bit of how we manipulate path length. So I'll give you an overview, and then Albert will get into more computational details on the actual impact of all of these phenomena. So the first way to manipulate path length is optical wavelength. I think intuitively everybody is familiar with this. You know that if you put blue light, so this is a rabbit ear with a tumor, and here is a transmission photograph using blue light, and all the light is sucked up by the tumor. But you do actually see contrast with these vessels surrounding here. But there's not that much light that comes through. So obviously the path length is very short. And if you look at the path length here in the blue and the green, where the absorption lengths and scattering lengths are about equal, they may be a few millimeters. And then as you move out beyond the green and into the red, so here in the red you don't see those small vessels. That big dark shadow is starting to diminish and some light is coming through. So obviously the light is traveling further. And as you move from the upper 500s into 650, 700, path length can increase by 20 or 30 fold. So there's this huge dynamic change in path length. So people are using this dynamic fluctuation in path length as a way to help localize light in space and do tomography. So that's one thing that you could do. Another thing that you can do is measure it explicitly so that you can then determine absorption and scattering. So this is the behavior in the scatter-dominated region, and this is the behavior where absorption and scattering are equal. And there is a tremendous dynamic as you undergo that transition. So definitely I encourage, if you're working in this regime, to figure out how to take advantage of this phenomenon, this nonlinear behavior. Now, even in this regime where there's quite a lot of light and the path lengths are very long, in the scatter-dominated regime, small changes in absorption can influence path length. So this is an illustration where we're going from 50% saturation. Saturation is oxygenated hemoglobin divided by total hemoglobin. So this is a consequence of metabolism. 50% saturation, there's a lot of oxygen extraction. 100% saturation, there's not as much extraction by the tissue. So let's just say a tissue is undergoing a metabolic event where there's a huge amount of extraction. And as you go from 100 to 50 or 50 to 100, you see that the path length can change by another factor of 2. So that's important. It comes into play a lot in neuroimaging studies where under stimulation the saturation may change and the path length may change under some type of metabolic perturbation in the tissue. So here's another personal view of path length. I like to show some of these pictures of myself as a way of illustrating the physical concepts in the field. So this is me illuminated at 400 nanometers. So remember, this is very low path length light. It's going to be on the order of a millimeter. You can think of the path length here as the penetration depth, more or less. So penetration depth is very shallow here at 400. And at 850 nanometers, where absorption lengths are much, much longer, much greater than scattering lengths, the penetration depth is much greater. So it influences contrast. So where I have very shallow penetrance, I'm seeing contrast from something that's entirely different when I have deeper penetration and longer path length light. So what's the contrast that you're seeing here with essentially invisible light? It's really hard to see blue light in that region. Why can you see all of those structures? Whatever they are, what are those things?
Like freckles, right, or something.
What are those? Melanin. Where is the melanin? Where exactly? Right, at the dermal epidermal junction in the basal layer. And most of it is localized in the basal layer and in some of the keratinocytes above the basal layer. So melanin is contained in melanosomes, granules that are about 1 micron, like the magic size. And they're taken up by keratinocytes, they're produced by melanocytes, so they're in melanocytes as well. But where are those structures? So we talked about they're at the dermal epidermal junction, but how deep is that? Is that 1 millimeter, 100 microns, 50 microns, 10 microns? How many say 1 millimeter? How many say 100 microns? How many say 50 microns? How many say 10 microns? So I can tell you, because I've measured this in myself, Vossen and Carol have these images. So my basal layer, the thickness of my epidermis is about 40 to 50 microns. So all of my melanin-containing cells, all the keratinocytes that have melanin, and my basal cells are like no greater than 50 microns deep. So all this contrast is coming from, when we talk about a superficial layer, that's a superficial layer. It's pretty amazing. Now, this image, basically the light just blasts through all of that. It's low energy, no interactions or very little interaction with the melanin, multiple light scattering, and I think I look a lot better here. All right, so let's say you are, now I just show you these two images and you get a job in a cosmetic company, you're a bioengineer in a cosmetic company, and they just hired you because they want you to make people look better. What are you going to do? You make all the room lights. You give it, everyone has to wear 850 nanometer viewing goggles. That sounds like a brute force technique. What could you do that's maybe a little bit more?
Make it more scattering in the blue, I guess.
How are you going to do that? How are you going to make everybody more scattering?
Make the particles a specific size.
Exactly. And there you have the secret of cosmetics. That's it. You'll make particles in your cosmetics that scatter light multiply, and it gives you a softer glow. But I still think everybody should wear 850 nanometer sensitive. That probably is the ultimate brute force. That'd be the real hardcore engineering way to do it. Okay, the second way to control path length, and you've heard about this and you'll hear a lot more about it tomorrow, is in space. So here, as I mentioned, absorption lengths in the near infrared are greater than scattering lengths. So typically they're about 10 centimeters. I don't know if I gave you a number, but you can sort of think of absorption lengths in the near infrared incredibly long. This is the average distance between absorption events. It's not the average distance between molecules. So the molecular distance is the same regardless of whether you're in the near infrared or in the visible. It's just that the probability of absorption is much lower in the near infrared. So the effective absorption length is greater. The scattering length is about 20 to 40 microns. So these are obviously on totally different spatial scales. So if I look at the reflectance as a function of source detector separation, I launch a source into tissue and I have a lot of detectors. Then I'll see this behavior that we like to call R of rho, the reflectance as a function of source detector separation. And we see that we have initially very rapid fall off in this region where the propagation of light is dominated by scattering. We say it's scatter dominated because scattering lengths are very short. So I'm really not affected much, or my photons are not affected much by absorption loss. And that's close to the source. And then, as you get further and further away from the source, you have a combination of absorption and scattering that are influencing the decay of the reflectance. And Albert will also go into more detail on this. But the key thing is that if I can measure close to the source and far from the source, then I can resolve absorption from scattering, because mathematically the behavior is different in these different regimes. And so Albert has some really more detail, interesting modeling detail on that. And the third way, we talked about wavelength, we talked about space, and the only dimension left is time. So if you take the absorption lengths and you divide them by the velocity of light, you have an absorption relaxation time. And this works out to on the order of tenths of nanoseconds, about a half of a nanosecond in tissue. Same thing true with the scattering lengths. Divide that by the velocity of light, you have a scattering relaxation time. So these are quite literally the average time between absorption events, the average time between scattering events. And time-resolved measurements are common in spectroscopy. And even though these things are pretty fast, a half a nanosecond and fractions of picoseconds, for optics and optoelectronics, that's not very tough. We can resolve phenomena on that time scale. So the first work that was done in this area was done by a group at Penn, and almost around the same time at University College London. At Penn, it was Britt Chance and his colleagues. University College London, it was David Delpy and his colleagues. So they were looking at pulses of light going into tissue. And what they saw was that if you took an ultra-fast laser pulse, a few picoseconds, and you put it into tissue, it would spread out in time. And that spreading in time is because of the fact that in that pulse, the photons are all traveling at the velocity of light in that pulse, but they take different paths. So some take longer to come out of the tissue. They take longer paths. Some come out very quickly. They emerge early. So the early emerging photons are the ones that are dominated by scattering. They haven't traveled as far. The later emerging photons are the ones that have been influenced by scattering and absorption. They've traveled further. They've undergone interactions with deeper structures. So you can sort of segment this, and Albert will also talk in a lot more detail, where you see these the early light and the later light contain different information content. If we fit the entire shape of the pulse to a model that comes from transport theory to how that should look, then we can deduce what scattering and absorption is. And so this is kind of just a cartoon illustration where you can see that the early emerging photons are really penetrating more superficially, and the later ones are integrating over a deeper volume. But there is very, very real and quite dramatic temporal dispersion. And your ability to measure this really depends on the sensitivity of your detection system. So you can imagine that if I keep waiting longer and longer times, there are probably not that many photons available. So people often use a technique called time-correlated single-photon counting to be able to measure these few photons that are arriving at later times. There are a lot of them around here, but if you want to go deeper and deeper and deeper in tissue, there are just fewer available. So to kind of summarize, we talked about these are techniques in the real domain where you've got some input function either in time or in space, constrained in time or space. And we'll measure some dispersion. So in time, that dispersion is temporal. We're looking at the spreading of a pulse. In space, we put in a dot. That could be from a fiber optic, or it could be just like this laser pointer. You can shine it onto the tissue, and you can then look at that dispersion of light, how it spreads out in space. And so those are real domain techniques. We work a lot in the frequency domain for a lot of different reasons, where instead of taking a delta function in time or a short pulse in time, we can modulate light in time at a single frequency or multiple frequencies. Or we can modulate light in space at a single frequency or multiple frequencies. So this pulse contains essentially lots of frequencies. So if we think of this time-modulated frequency domain approach, it's like a monochromatic approach. This pulse contains all the frequencies that we're going to need to describe the tissue. But for some practical reasons, we may not need an infinite amount of frequency content. We may only need a few frequencies to describe the tissue. So now I can do a kind of a monochromatic experiment by illuminating the tissue at a single frequency or a few frequencies and watch how these intensity modulated waves propagate through the tissue. And these are called, we call these photon density waves. So that's a mathematical and a physical construct, photon density waves. I'll talk about those in a second, but let me just introduce you to the instrumentation that we use to measure the temporally modulated waves. Tomorrow you'll hear more about the spatially modulated waves, but the conceptual framework is the same. So temporal modulation involves literally turning on and off a light source. And we start with a laser diode. So actually you can use any light source, but we need to get into the megahertz to gigahertz regime. Will this technique work in the kilohertz? I just kind of gave you a hint on that. How many say no? No. What? How come? Well, so just to sort of rearrange your answer, it's not those are not fast enough waves to be sensitive to the dispersion that we need to measure. So they're not. Those waves are interacting with the material, and I'll get into that in just another slide. But if you're just modulating in the hertz or kilohertz regime, if you take the reciprocal of those frequencies, then those relaxation times are too slow. We need to be modulating on the order of the relaxation time of the tissue. What influences the relaxation time of the tissue is the absorption relaxation time and the scattering relaxation time. So we need to be within or around the lowest one, which is the absorption relaxation time, which is on the order of half a nanosecond. So we need to be using waves that are fast enough to be probing processes on that time scale. So we're going to modulate light in the megahertz to gigahertz regime because that's going to be fast enough to interrogate to get a response from the tissue. Are any of you guys modulating light? Intensity modulating light? How do you do it?
Well, we actually use one of the MI systems.
Okay, but that's in space.
Yeah.
So how about in time? Albert, are you going to say anything about the instrumentation? Because it's interesting how you do it. I'll say one quick thing. I mean, we use laser diodes, but what's important is how many of you have heard of a bias T?
I don't quite remember what it does, though.
So it's a little mixing circuit that allows you to mix AC and DC together. So why is that important with a laser diode? So laser diodes all have threshold current levels that you need to hit. What if you're not at that threshold level? What do you have? You have a very expensive LED. Well, probably not very expensive. It's not as expensive as it used to be. But you basically have an LED. So the difference between the LED and the LD is you've got a cavity structure, first of all, that's conducive to lasing. But you've given just enough current to get above threshold for that lasing process to occur. Now, if you just get right above that threshold, so you're just, lasing, and then you hit it with RF, so now you're swinging the current very fast in time, you will get the light output to follow the current. So that's the trick with modulating laser diodes. Now, of course, it's very simple, but I think my first NIH grant was basically how we were going to modulate laser diodes at frequencies in the megahertz to gigahertz regime. And there were a lot of difficulties, and the laser diodes that we were buying from Spectra Diode Labs, which was a spin-off from Spectra Physics, cost thousands of dollars. And then we had to get a special collaboration with a guy at University of Wisconsin who made these things in his laboratory. Now, of course, you can just buy them anywhere. And they are super inexpensive. But it's still somewhat of an art form to modulate these sources in time. So what we have to do is we have to take the laser diode. We have to use a special little mixing circuit called a bias T. We bring it above threshold, and we hit it with RF. If we hit it with too much RF, you can imagine that there's this current power relationship between the laser diode. If I bring it up above threshold, I'm going to get lasing. I can keep giving it current, and I'll keep getting lasing. But if I give it too much current, then the system collapses, and I get no more lasing. Things are clipping. So I can't have RF with amplitudes that are too great, or else I'll get nonlinear harmonics generated in my laser, and everything will be really weird. So I have to make sure that the RF amplitude is just the right amplitude so that I'm getting very beautiful sinusoidal modulation. And so you can tune your lasers with that. So we'll get this modulation, and we will get propagation of these waves into tissue. So this is a scalar photon density wave. I maintain the modulation characteristics in time. And as they go through tissue, they undergo dispersion. Dispersion means that the phase velocity of the wave is nonlinear with frequency in some way. And we'll talk a little bit about that. And then I'll have a detector that's collecting some of the remitted, the backscattered light, and I measure the phase and the amplitude compared to a reference signal. And from that phase and amplitude, literally, here's the measurement, and I'll fit that to the model, which Albert will tell you about. And I can deduce then the absorption and scattering properties of the tissue. So what must that model contain in order for me to get those fits? What are the components in the model that are important? I actually just told you. So this is a model-based fit. The dots are the measured points, and then the lines are the fit to the model. So the model, which is derived from the transport equation and a diffusion approximation, if you look at the terms like phase equals, amplitude equals, in there are absorption and scattering. So it's a nonlinear model. So I have to have some nonlinear behavior in order to get separation of absorption from scattering. So that's really important. So this is a big issue in inverse problems. If you have nonlinear models, you need to make sure that your experiments have sufficient nonlinearity in them so that you can adequately resolve things. What happens if you can't adequately resolve free parameters in your model? What's that situation called? Ill-posed, ill-conditioned, you're screwed. Do another experiment. Make another measurement. Try to get orthogonal information content. Try to get a better model. So this is the general map, the roadmap of a photon density wave. And I'm not going to go into a lot of detail on this, but what I do want to emphasize, and Albert will mention more of this, is that this is a scalar wave that you can describe using wave characteristics. So it's got real and imaginary components. And we typically, so due to formalism, we describe the phase in terms of the k imaginary, the complex wave number, the imaginary component of the complex wave number. And we describe the amplitude in terms of the real component of the wave number. And as we go to higher and higher frequencies, ki and kr get closer and closer together, or approximately the same. And as we go to lower and lower frequencies, you see that the phase actually becomes linear with frequency at low frequencies and nonlinear at high frequencies. And the amplitude, the kr, just drops off. And the limiting factor in kr is the DC penetration depth. So at low frequencies, you're just looking at DC light propagation. And the penetration depth of that intensity modulated wave is the same thing as the DC penetration depth. And then as we go to higher and higher frequencies, the penetration depth is given by 1 over kr. So I can limit the penetration depth. So this is the same thing. What's another technique where if you increase the frequency, you don't have deep penetrance? A common medical imaging technique that everybody uses, that is handheld and is in real time. Ultrasound. So what is that thing in your hand called? A transducer. And how do I get measurements of skin versus measurements of, let's say, breast tumors? Do I use the same transducer? What's the difference between the transducers? Okay, you're a biomedical engineer developing ultrasound transducers. What are you going to do to see skin? How will you tune your transducer? Higher frequencies. They don't penetrate as deeply, and you'll hear a lot more about that from Sylvain tomorrow. Lower frequencies, they will go deeper. And here it is in complex wave numbers of photon density waves. Higher frequencies don't have as deep a penetrance as low frequencies. In fact, at the limit of low frequency, the real part of the complex wave number is just the DC penetration depth. And another interesting feature to this is that the phase velocity of the wave, at low frequencies, the phase velocity is constant because that's basically omega over k, so it's linear. But at higher frequencies, as we're up here, it's proportional to omega to the 1/2, the square root of omega. And that's where we get that dispersion. That's that nonlinear propagation. And then if we look at the wavelength of a photon density wave, it seems pretty long. So at 200 megahertz, it's about 10 centimeters for these optical properties. That's 10 centimeters. Now, what is that with respect to the absorption length? Is it the same as the absorption length, a lot shorter than the absorption length, or bigger than the absorption length? Well, in this case, I actually gave you the optical properties mu sub a. That tells you what the absorption length is. So in this case, this is just a fraction of the absorption length. So it's almost like a near field technique. You're looking in the near field. An absorption event is going to occur further away than the photon density wavelength. But that wavelength, is that shorter than the wavelength of that photon density wave in air, in a non-scattering medium, or longer? So here's your answer. It's on the board. So if I take that RF modulated wave and I just launch it in a fiber or in air, it's meters, 10 meters, 9 meters. This one's 9 meters. So why is it so much shorter? Scattering. Multiple light scattering slows the wave down, and it changes the wavelength. Okay, so we talked about these scalar density waves. You'll hear about spatial modulation tomorrow. The whole trick is to measure some MTF with frequency. If you have fibers that are placed on the tissue, you can measure a depth, like 8, 10 centimeters in a transillumination geometry. In a reflectance geometry, you're not measuring as deeply, but you can measure certainly several centimeters. You can go through skull and into brain. And in reflectance geometry, you're typically looking at several millimeters, possibly up to a centimeter, but sort of the sweet spot is in about 5 millimeters or so. So what's the commercial or clinical status of all this stuff? So there are more than 20 near-infrared oximetry companies. And so this is, I guess, compiled from, I think we're not constantly updating this table, but this is more or less within the last year or so. And they kind of work out to be point probes and imagers. And there's really a good article by Claire Elwell and Chris Cooper that kind of summarizes some of these. And the point probes, some of them are time domain and frequency domain. So this ISS probe, Hamamatsu. But you can see there are hardly any time domain or frequency domain devices that are commercially available. Very, very few. So that should tell you something. One thing it says is that it's relatively easy to make a CW device. There are business models for that with disposable elements. And it also tells you that maybe we should be looking at all of these things because there are 8 that are FDA approved. So this is moving pretty fast. People talk a lot about how fast the OCT companies are moving. This is also moving very fast with many companies that are developing systems that are FDA approved. There aren't the same really nice high-resolved anatomic images, but they do go into the clinic and they are making measurements in patients and influencing clinical care. So here are some of the form factors in these. This one, the InfraScan, was recently approved about a year and a half ago, which it's designed to measure traumatic brain injury, and it's actually being used close to us in Camp Pendleton by the Marines, and it's kind of like a stud sensor for the brain. So it's really nicely engineered, little handheld device. You put it on one side and compare it to the other side, and you can see if there's any subdural hematoma formation due to a traumatic injury. So if you have a traumatic injury to the skull, to the head, whether it's a blast wave or a blunt injury, or even a projectile injury, the amplitude of the shock wave is greatest at the outside or right at the interface between the hard skull and the soft gelatinous tissue. So in some sense, that's really nicely structured for optical techniques. They will not look at deeper structure damage, but you can deduce whether or not there is hemorrhage at that interface where the amplitude of that wave is greatest. And these are all being used mostly for brain monitoring, many of these things for operative stuff. So here's an example of something in the literature we didn't do, it didn't come from our community. This is a group of anesthesiologists in Germany, in Lübeck, where they took that Covidian device, the Somnetics oximeter, and they wanted to use it in patients who were receiving heart bypass surgery. So you can imagine if you're having bypass, that's a pretty traumatic procedure, and you would like to know what's going on in the patient's brain when they go on bypass. That's a potential risk. But what these guys did was they said, what is your brain saturation even before we start bypass? Because there is an outstanding question, and that is, are you a good candidate for surgery? Are you going to come through the surgery? So the only question that this group was asking in this study of 1,200 patients was, is your brain saturation high enough before you go into surgery so that you will have a good outcome after surgery, assuming that the surgery is going to be good? Could your brain saturation be a limiting factor in your survival after surgery? And what they found was if they set a cutoff of 50% saturation, that you only had about a 70% or so chance of survival at 1 year. Whereas if your saturation was above 50%, you had a 95% chance of survival at 1 year. Now that's a huge difference. This is looking at all 1,200 subjects. And then if they further stratified these populations into a high and low risk group, you had high blood pressure, you were obese, you had diabetes, so these were all high risk people. The difference between that group, 45% were alive at 1 year versus about 80% or so if you were not in the high risk group. So this is a really classic example of what type of study that I talked about earlier on that we should all aspire to at least once or twice. Starts with an O, an outcomes-based study. Not designed by anybody from the optics community, designed by physicians. Really interesting. But I think in a lot of ways, it's great for us. It raises a lot of questions, too. What if your saturation was 53%? 57%? You know, that's most of the people. That 50% cutoff is really extreme. I mean, it's possible that, I don't know, I don't think you could just tell by looking at people whether their saturations are 50% or lower, but that's a pretty low saturation. So we need to be able to get numbers that are more quantitative. Maybe relative saturation is not quite good enough. We need to be looking at the majority of the population that are in between 50 and 100% and do better for those people. That's really most people. So what we believe is that quantitative measurements, more quantitative measurements could be used here and improve outcome. And there may be other optical endpoints. So we've got all that spectral content. There may be endpoints that could be just as powerful or even more powerful. Maybe we could use these endpoints dynamically during surgery, and we could use that to make clinical decisions. So we've been involved with Lingzhong Meng, who's an anesthesiologist who was here for a while, and now he's moved to UC San Francisco, and we're still working with him. And he's put lots of patients on protocol using an ISS device. This is a frequency domain device, and you'll see it this afternoon. And so Lingzhong actually came over to BLI, and we were talking, and it's shocking what anesthesiologists don't know. So the first thing they don't know is, do their patients feel any pain? They have no idea. So there's a lot of work going on in our field now to try to use these techniques to assess whether patients actually feel pain during surgery. The second is, they've got a pulse ox. Where is the pulse oximeter on every patient? On their fingertip. And everyone assumes that the arterial saturation in your fingertip is proportional to the tissue saturation, the oxygen utilization, and metabolism in your brain. And it is, but it's a very weak relationship. So by the time your arterial saturation in your fingertip, the alarm is going off and it's crashing, probably you've had a pretty prolonged ischemic event in the brain. So here's a study that actually kind of looks at that. Although nothing happened in the fingertip pulse ox. So I'll just kind of run you through this. When you get put in under anesthesia, so the first thing that happens, what is the first thing that happens when they put you under? What's the major physiologic consequence of going under anesthesia? How did Michael Jackson die? What did they give him? Propofol. What happened? Why did he die? What's the biggest risk of anesthesia? Everything gets suppressed. Breathing, what else? Heart, cardiac output. So what's going to happen to your blood pressure? Goes way down. Your blood pressure drops. So the first thing that an anesthesiologist has to do is get your pressure back up. So how do they do that? What technique? What do you have at your disposal to get pressure back up? Drugs? Did you say drugs? Okay, what are the drugs going to do? Peripheral vascular, yeah. So they're vasopressors, vasoconstrictors. They increase, or they press against your peripheral vasculature, your peripheral vasculature is undergoing a constriction, and so that allows you to increase your central pressure. So your mean arterial pressure goes up. So here are two different drugs that are commonly used, phenylephrine and ephedrine. Have you heard of these drugs? No one's heard of ephedrine? Ephedrine is, pretty commonly used over the counter. Well, it's actually a vasopressor, but it's not an antihistamine. It's a decongestant. So ephedrine and pseudoephedrine, and you can't really buy them because, in abundance anyway, because what can you do with them? Yeah. So here, it's widely used. This is an old drug. So what you see is your pressure drops, very low mean arterial pressures. You give the drug, and you see an increase in the mean arterial pressure. So with phenylephrine, it actually goes up pretty fast. And ephedrine, it's a slower climb. So what is going on then in your brain? So this is the measurement of saturation in your brain as they give the bolus of these pressors. So this is comparing phenylephrine to ephedrine in two different patients. But this was, I forget how many patients, 40 patients or something? 30 or 40 patients. So what happens is your saturation in your brain goes down. It goes down with the phenylephrine, but it's really flat with the ephedrine. So there's really no change in brain saturation. So it drops a few percent, and then it climbs back up. Why does it climb back up? Well, they're giving a bolus. So they give a little bit, and then the effect wears off, and then they have to give more. So if you're under anesthesia for a long time, you get this, it goes back, then you get another one, it goes back, and another one, it goes back. So your oxygenation, your brain oxygenation will go down and come back, and down and come back. So it depends on the frequency of the injection. What if they're giving lots of injections back to back to back? They may not actually ever see that you recover. And what is the impact of these desaturations? Nobody really knows, but there's been work also to look at postoperative cognitive decline after surgery. And they've correlated these ischemic events during surgery to impaired scores in this mini mental task test that tests your postoperative cognitive abilities. So actually it's a real problem that people encounter in surgery. Why does the phenylephrine versus the ephedrine, why are they different? It's probably due ultimately to the modulation of these effects on your mean arterial pressure. So this is a more gradual mean arterial pressure modulator. This is a more acute one. So if suddenly your mean arterial pressure is going up a lot, then your cardiac output has to compensate. Otherwise, you will blow up. So your cardiac output will drop. And they have you monitored with a lot of this stuff, blood flow, cardiac output. They're looking at all that, but it seems to me they're missing one really important piece of the puzzle, which is what's going on in your brain. Because cerebral autoregulation of flow is extremely important, and it will be influenced by all of these other factors that are going on. So this is a way of accessing that in patients undergoing anesthesia. And I think that this type of work combined with the previous study by the group in Germany, it will be just a matter of time before there will be lawsuits, lawyers will get a hold of all that, and I think that everybody should be wearing those things in some form or another. All right, so here's some other kind of interesting and fun studies. We're very, very interested in your brain and muscle during exercise. And with changes in healthcare, I guess all of you guys are following that. Healthcare reform, high cost of healthcare, diminished Medicaid and Medicare reimbursements. So basically, healthcare is pretty much up to you. You're on your own. No one's going to be able to afford drugs or expensive imaging devices. So the best thing that you can do is exercise. It's the best drug. We're looking at it as a way of treating diabetes, metabolic syndrome, and obesity in kids. And so some of the first things that we have to do is sort of get a baseline of what's going on when you exercise. And then if you give an exercise dose, what's going on in your brain, what's going on in your muscle. So these are some interesting studies that we're doing. You'll see this instrument as well. This is a time-resolved system by Hamamatsu. And we've got a probe on the vastus lateralis muscle. We've got a probe in prefrontal cortex. And so this is Warren, who played football at Stanford. And now he's in grad school. Hopefully will come back and work with us next year. But he's been a big leader in these studies. We're also working with Gautam Ganesan, who's the MD PhD student, who's really taken these studies off and is very focused on using exercise as a serious intervention in healthcare. So basically, we want to look at your brain and your muscle and how they work together during exercise and get some baselines of what's going on. So this is what's called a cycling ergometer. And what it does is there's an increase in resistance, 20 watts per minute, and it's ramped up until you're exhausted and can't go any further. So this is not Warren. This is another subject. This is actually this is not an adult. This is a kid because we're looking at kids. So this is the brain tracing, brain tissue oxygenation in the frontal cortex. And this is the muscle. So first in the muscle, what you see, this is rest, just pedaling in an unloaded way. Here's the ramped exercise. And you'll see oxygen extraction by the muscle. Saturation drops because the muscle is working. And we call this building up an oxygen debt. That's repaid to the muscle in recovery. So as soon as you stop, there's reperfusion to the site, and you get reoxygenation. So that's kind of what you would see in a working setting for a muscle. That's very typical. What's going on in the brain? So everything looks pretty good in brain saturation until here or so, and then you start to see this drop. Saturation drops pretty significantly, and then you stop, and it stays low. So we thought this is a pretty interesting observation and wondered whether or not this desaturation, this is not motor cortex. This is prefrontal cortex. It's an executive function. Maybe this is focus, maybe it's fatigue. We don't really know what the origin of that signal is. So if you look at the muscle separately, and this is deoxyhemoglobin, and this is oxyhemoglobin, what you see is that during exercise, there's an increased rate of production or an increased production of deoxyhemoglobin that begins to level off. That deoxyhemoglobin increase is occurring at the expense of oxyhemoglobin. So you're pulling oxygen off as a result of cellular metabolism in the muscle. Muscle's working. But here, you start to plateau. So what's this phase probably called? You probably all have been there. What are you making a lot of? Lactic acid. Yeah. So the muscle can't use oxygen anymore. It's super fatigued. You're not able to use oxygen. You're using glucose as a substrate, or whatever you can get the cells can get their hands on as a substrate, and then building up lactate. And it's really evidenced beautifully in these signals. And then you stop, oxyhemoglobin goes up, massive reperfusion paying back that oxygen debt. And why does deoxyhemoglobin go down? So quickly like that. Think of plumbing. all that oxygenated blood comes rushing in, and it dilutes out the deoxyhemoglobin. So it's like a plumbing effect. So this is a classic muscle response. What's really beautiful is we can separate oxidative from glycolytic metabolism. We don't know exactly when the threshold is for the glycolytic metabolism. It's clearly before this point. So it's probably earlier on. We can get even more clues if we look at the brain. So this is oxy and deoxyhemoglobin in brain. And you see they're fine as you start to pedal. And you start to see this rapid rise in oxyhemoglobin into the brain. And then you hit some probably respiratory threshold here, and you start to see this increase in deoxyhemoglobin as well. So both oxy and deoxy are both going up over some interval at the same time. So there's metabolism and there's perfusion. So why do you think the oxyhemoglobin is going up? In the brain. You're not working your brain like you're working your muscle. Well, you're making ATP. And what else is made when you make ATP? What's a big end product of ATP? So you're breathing, you're taking in air, and what comes out? CO2. Why does CO2 come out? I mean, it's always coming out. Why is it coming out? Where is the CO2 coming from? Where is the CO2 made? Like, how did you get it in the first place? Take a guess. Well, a pretty good answer in this lecture is mitochondria. So it's always a good answer. Scattering and mitochondria are always like, one of the two is going to be right. So basically, you've got oxidative phosphorylation going on, right? And so one of the products of that is ATP and CO2. So CO2 then ends up in the vasculature and is exchanged in the lungs. And we have a sensor that's measuring what's called end-tidal CO2. So normally there's some concentration of CO2 in your breath. And then it's going to increase as you undergo strenuous exercise because you're going to be making more ATP and CO2, so you get more end-tidal CO2. Now, if there's more CO2 in your blood, what happens to the pH of your blood? It does go up or down. But it goes. Your second answer is right. It goes down. So you get carbonic acid formed. So what does your body do to accommodate that? You try to get rid of it. And what else? What does your body do to the blood? What does your body think is going on if your CO2 levels in your blood are going up? Thinks you're suffocating. Who said that? All right, good, a physiologist. So it thinks you're suffocating. So if you're suffocating, you're not getting enough oxygen to your tissues, what is the vascular response going to be? Vasodilation. And so that's what's going on here. So your brain thinks it's suffocating, and you'll get vasodilation, enhanced perfusion to the brain. This happens to be a time when end-tidal CO2 is going up. Now, here it flips, and it's coming down. But the deoxyhemoglobin continues to go up. And actually, deoxyhemoglobin is persistently elevated. And we're not exactly sure why, but what we think here is that the blood supply can't meet the metabolic demand at this point. So the body thinks it's suffocating, it's increasing the perfusion, but there's still demand by the cortex Maybe the cortex is using the lactate, because now we know from the muscle studies that lactate is building up, so lactate can be used as a substrate. And it may be that the cortex is using that very actively, the pH is changing, and we see this continued increase in deoxyhemoglobin, even when we have this flip. So somehow there's still metabolic activity going on in the cortex. So these are really interesting kinds of concepts that you can explore with these technologies. It looks like, you know, this may be a state of exhaustion, which you guys are probably in. And I have so much more to tell you. That's why we have 4-hour summer schools in Sweden, or 3.5 hours or so. And these are the end-tidal CO2 tracings for that individual. So you'll see end-tidal CO2 going up and then coming down, mirroring that oxyhemoglobin change. And we found that this drop in particular is very, very highly correlated with prefrontal cortex oxyhemoglobin levels. So maybe a good way of measuring end-tidal CO2. This is Warren's brother, Conrad, and he plays for the Jets. And he has set the record for this biodex dynamometer in terms of the amount of torque that he's able to exert. I'm going to skip over this. This is interesting. Everybody wants to know how to build muscle, and I'll just leave you with that. We have some insight into how to do that with reduced load on the muscle, which we think is important for rehabilitation, particularly if you have injuries to joints, aging, and so forth. But these are the types of studies that you can do with that. And so now I want to introduce, so those were oxy and deoxyhemoglobin studies. There's a lot more information content from wavelengths in between where the laser diodes are. We want to get to that information content. And one way to do that is to combine laser diodes with broadband or white light techniques. These can be time-independent methods, and these are time-dependent methods. And what we do is, with the time-dependent, the frequency domain methods, we measure mu sub s prime at a few wavelengths explicitly. DC reflectance is uncalibrated. It can vary depending upon the contact you have with the tissue. It's just a DC technique, and you don't know what the path length is and all that stuff. But if you know what the wavelength dependence of scattering is, and we know that this falls off in this sort of simple type of behavior, a lambda to the minus b, then we can force the DC reflectance spectra to fall on top of this line, and then convolve that with the spectrum. And then what falls out mathematically is the absorption spectrum. So we force that DC reflectance, which is coming from non-modulated sources, to behave like this. What is that called mathematically in an inverse problem that begins with a C? It's a constraint. I'm constraining, I have some prior knowledge of the system. I know that the system should have scattering behavior like a lambda to the minus b. I'm going to explicitly get that at a few wavelengths using frequency domain techniques which separate absorption from scattering. Now I can just take some DC wavelengths, make them sit on top of that, and then the rest of the reflectance component has to be due to absorption. And so this is a way that we can get a complete absorption and scattering spectrum. And we can build it into a probe. And as we take that probe and move it across a tissue, we'll see the absorption spectrum change. So no longer are we just looking at oxy and deoxyhemoglobin. Now we're looking at water and lipid and unknown components. Because even though we say that, you know, most of the tissue is oxy, deoxy, water, and lipid, it's got other stuff in it. We kind of have an idea of what the other stuff is, but we don't know for sure. And I won't go into this, but a big area of work that's gone on in our lab and others is to see what the difference is between the known components and the unknown world. And those are techniques that are based on looking at different spectra or residual analysis. But just from this spectra, we get a lot of information now about important contrast elements in tissue, water, lipid, oxy, and deoxy. And by having this constraint, we can improve the accuracy of our fits. And so with this approach, we've done a lot of work in breast cancer, and we've done detection in dense breast tissue and guiding therapies, neoadjuvant chemotherapy. And I'll just give you a few more slides because then I'll stop at 10:30 and. Okay, I'll try to. Well, maybe I'll accelerate through this. So the functional contrast in breast, as you might imagine, comes from there's more blood in tumors. So tumors have more vascular supply. You hear of angiogenesis. This is a direct measure of angiogenesis. There's a change in metabolism in the tumor, and that's detected by changes in the hemoglobin content, total oxy and deoxy. And there's more water in tumor. So the water is a couple of factors. First, what's the cell density in a tumor versus a normal tissue? Is it the same? Is it higher? Is it lower? Higher. By a factor of 2, 5, 10, or 100. 10? 10? How many vote for 10? How many vote for 5? How many vote for 100? Okay, it's a good distribution. It's about 10. It's about an order of magnitude higher. What is a cell made of? Mostly. Water. What's the body made of mostly? Water. What's the planet made of mostly? So those proportions are all about the same. So mostly water. You have an order of magnitude more water or more cells, so you got more water. What's another factor that drives water into tumors? How many of you heard of the EPR effect? What's it stand for? Enhanced permeability and retention by vasculature. So how does that work with water? Where do people take advantage of that? How do people take advantage of that in cancer?
For when they do nanoparticles, they introduce it systemically, and then because of the EPR, the tumors are really leaky, so they naturally go there and leak out.
Right, exactly. So you can deliver stuff to tumors. But let's say you're not delivering anything. Let's say the tumor is growing. When we talk about leaky vasculature, from a physical point of view, those are literally gaps between endothelial cells in the vasculature. So what is normally leaking out of the vasculature as a tumor grows? What's the biggest leaker? Where's the biggest... I guess it's not wiki leak, it's tumor leak. Water. Add that to the third answer. Mitochondria, water, oxygen. So water extravasates. And that leads to the edematous response. Where else do you get edematous response? In wounds, so injuries where you're crushing vasculature, in inflammation. So if you have an inflammatory response that's occurring as a result of an allergic reaction, you get swelling. That's edema. That's tissue edema. So water shifts, water movement. Don't underestimate the importance of that, both from a physiologic point of view as well as from a measurement opportunity point of view. Tumors also, as they develop, they displace bulk lipid. So breast tissue is constructed with glandular structures that are literally embedded in a lipid protective cage. And as the tumor grows and begins in epithelial cells within those glandular structures, it begins to push that out as the solid tumor grows. So that's another contrast element that's available to us. So there's typically decreased lipid. And this is what it looks like. So here's a young woman with about a 3 centimeter mass. And this is the left side without the tumor, oxyhemoglobin, deoxy, water, lipid, and then what we call a tissue optical index, which is where we've just combined deoxyhemoglobin, water, which go up in tumors, and lipid, which goes down. It goes in the denominator. So it's a high contrast index. And you see, this is the right side where the tumor is. You can see an increase in oxy, a localized increase in deoxy. There's more water, there's reduced lipid, and then our index can range from a factor of 2 to 20-fold contrast. And that's just based on endogenous molecules, endogenous contrast. So there's a lot of endogenous contrast that's available. And one of the things that we're trying to do with this is measure these signatures before chemotherapy, look at them, their spectra and the images, and you can see the scattering and absorption spectra look quite different between tumor and normal tissue. And then follow patients as they get chemotherapy. So this tumor probably took 10 or 15 years to grow. And then they get chemotherapy, and in 6 months it may go away. So there's a lot of really dynamic physiology. And what everybody is trying to do in the imaging world is measure at different time intervals and predict whether or not this patient, who will then go on to surgery, and a pathologist will look at the specimen and say, this is a complete pathologic response, this is a partial pathologic response. If that pathologist says it's a complete response, that influences or will dictate outcome for that patient. So those patients that have a complete response have better 5-year survival than patients that don't have one. So this approach, what is called neoadjuvant chemotherapy, pre-surgical, is growing a lot in the breast cancer community, in colon cancer, in head and neck cancer, because oncologists can begin to see if a patient will respond to chemotherapy before they have surgery. That's important because if you get chemotherapy after surgery, how do you know if that patient is sensitive to that drug? So what do you think is the fraction of patients that don't respond to chemotherapy? How many? 70? That would be bad. It's about 20% or so. Patients don't respond at all. We don't know why. But if you want to know if that patient's going to be a responder, it's better to give them the chemotherapy while they have a tumor than after you've taken the tumor out and you have absolutely no way to trace their sensitivity to the drug. So that's why this is growing in popularity. And we kind of got lucky making these measurements in the early 2000s as people began to recognize this and epidemiologic studies came out showing the importance of having a response to pre-surgical chemotherapy. So we've been able to now do a multi-center trial using this technology, trying to predict that. We want to predict this as early as possible. Ideally, we'd like to predict it before they even start. But we want to predict it a day after they get chemo. And so I'm going to skip over all the work that we've done kind of leading up to just one key point. So these are images of a patient before chemotherapy. This is deoxyhemoglobin of their tumor. This is just 3 days after. And here is 1 week later. So this is the first infusion. So they sit in the infusion center. They get an infusion for several hours of chemotherapy. They came back 3 days later, and it looks like the tumor is going away. But in fact, the mass is still there. This is a metabolic. This is a functional response. There's less oxygen consumption. Deoxyhemoglobin has gone down. But here, 1 week later, it's way up again. We want to know from these the dynamics of this, is that patient going to be responsive? And in fact, if we sort of make a decision on day 3, we might predict that that will be a responder. But if we make a decision on day 7, we would say, well, the patient's not going to respond. And what's really interesting, and no one has seen this before because you can't really do this with PET, you can't do this with MRI, it's not practical, and you don't even get the same information content, that in that first week, you can get lots of fluctuation. And this is oxyhemoglobin for responders versus non-responders. And there may be information even on the first day. So we looked at this in a little bit more detail, and Darren Robillé and Shigeto Ueda led this study, and they showed, oops, this is backwards. I'll skip over this. What they showed was that the pathologic responders seemed to show an oxyhemoglobin flare on the first day. And if the patients had that flare, then they were likely to be responders to the chemotherapy. And that flare is really inflammation. So you're killing endothelial cells in the vasculature, you're also killing tumor cells, and the body responds to that. It responds with increased perfusion, enhanced vasodilation. And that's basically what we're measuring at the tumor. And if the tumor is able to demonstrate vascular reactivity, then it probably has vasculature that looks more like normal vasculature. So one of the hallmarks of tumor vasculature is it's not very reactive. It doesn't have smooth muscle cells. It doesn't have parasites. So it doesn't really expand and contract in response to a demand like normal tissue does. But that doesn't mean it doesn't have any of that stuff. And that also doesn't mean that it's not connected to normal vasculature. So this is a way of pinging or perturbing the tumor and then looking at the impact of that. And if the tumor does seem to have the capacity for reactive vasculature, it's more promising for it to respond. Why do you think that? What's the single biggest problem in the delivery of drugs to solid tumors? We have a lot of drugs that work pretty well on tumor cells, but why is it always complicated when they go into patients? Why don't they go to the tumor?
Why would they go? Why would they only go there to begin with?
Well, let's say the tumor has a 5 times higher blood volume fraction. So you get partitioning into a distribution of a molecule that's intravascularly injected in proportion to its blood volume fraction as compared to an adjacent tissue. What's wrong with the plumbing? There's high pressure. High osmotic pressures. Where does the high osmotic pressure come from? Extravasation of water and changes in osmotic character in the tumor compared to the surrounding tissue. It makes it hard. There's lots of back pressure, and it's hard to get molecules into it. So that's a big deal. Now, if you can assess whether the vasculature is sort of normal, is reactive, then you can get some peristaltic action that helps move molecules into the tumor. And you probably don't have high osmotic pressures. We haven't measured the osmotic pressures explicitly in proportion to the water content, but our assumption is that if there's lower water content, the osmotic pressures won't be as high. So I'm going to skip that. I'll skip all of this and sort of just say we've got a multi-center study where we're looking at outcomes, and we've delivered instruments all over the country, and we've finished enrolling 60 patients in this, and we're now waiting until all the patients finish their chemotherapy, which takes about 6 months for every patient. So it's a big job to measure all the patients for 6 months. and you'll see this in the lab this afternoon, that the technology, I started working on it in the early 90s, and it took up an entire room. And as all good things in our field go, they get smaller and less expensive. So here's the refrigerator size instrument, which you'll see. And here's the external hard drive size device that has 4 laser diodes in it and a board for driving the sources, and the APD is inside of this box, APD avalanche photodiode detector. You can take that box and put it in a bigger box that Amanda is doing here, and that contains also the spectrometer. So this mini system, which we'll sometimes call MDOSI, does exactly the same thing as one of these big refrigerator size devices that's based on a network analyzer. And we've even gone so far as to do whole breast mapping. So this gives us a totally different view of what's going on. This is a normal subject, and some of what we're trying to do is measure breast density. Breast density is related to risk. And we want to follow breast density as patients are undergoing therapies to change their density. So SERM agents, chemopreventative agents like tamoxifen. And so this is whole breast mapping. Here is the tissue optical index of an entire patient, and that can be now visualized in a 3D type of projection. And we can do the same thing with our tumor mapping. So here's a patient where you see the tumor before therapy, and here's at the midpoint, or actually this is the early therapy response. And you'll also see this afternoon. Here's an instrument that has multiple views. Now, it's not a classic tomography instrument where you have a ring or a planar geometry with hundreds of sources and detectors. We call this sparse tomography. It's rich in spectral content. So in every channel, there are 4 separate views, 4 separate channels. It has all the wavelengths and all the frequencies. And the goal here is to be able to reconstruct, much like we do with ultrasound, with a handheld probe. So, summarize. We can measure hemodynamics and composition with these methods, and they can be quite powerful. They're right at the bedside. We can localize inhomogeneities, measure perfusion and metabolism, fluorescence. Tumors are a good target. But as I was showing you with exercise and anesthesia, anywhere where you have cells and blood, you can do something that can be interesting. So a lot of times we try to look at perturbations. They may be an occlusion, they may be looking at a drug effect, they may be looking at radiation, or even breathing, and I think you'll see some of that this afternoon. And the bottom line is that these methods are good for individual physiology. We want to have more individualized medical care. Many people are trying to do this by looking at genetic analysis. That's pretty abstract. It's very upstream. It's tough to do. Our field is really, really good at looking at downstream effects, physiology, metabolism, composition, signatures that come from spectroscopy. And so these methods are very, very powerful for looking at that. And they're also more or less calibrated to the language of physicians. DOSI imaging team. You'll see a lot of people today, current members of the group, others who've been in the group and have worked on these technologies, and the Cancer Center folks. So you'll see that. We have support that we need to acknowledge as well from the NIH. So I think you're probably wiped out. And let's stop there.